Hello everyone, and welcome to today's The Scientist webinar. I'm Betsy Young, Interim Director of Creative Services for The Scientist, and I'll be moderating our discussion. Today, our panel of speakers will be sharing their research focused on cancer cell signaling. While their areas of study differ, one with a specialty in women's cancer, and the other interested in cancers of the central nervous system, they're both intent on understanding how cancer cells communicate and how we can interfere with that communication to therapeutic ends. We like our webinars to be interactive. We encourage you to send us your questions or comments at any point during the webinar, and the panelists will address these during the Q&A session following the presentations. To ask a question, simply type your query into the question box located on the bottom left-hand side of your screen. We will try to address as many of these questions as we can during our Q&A session. The webinar platform is user-friendly. You can move or resize any of the windows by simply grabbing them at the top or stretching them at the bottom right-hand corner. You may need to move or minimize some of the windows to see the live view. The webinar will be archived on the Scientist website, and we will send you the link via email within a couple of days. Please note, you will not be able to download the presentation slides. I'd like to take this opportunity to thank our webinar sponsors. Rockland Immunochemicals, Inc. offers each academic, biopharma, and diagnostic professional thousands of antibodies with the aim of providing the right antibody that is the perfect fit for every occasion. With facilities in Pennsylvania for over 50 years, Rockland manufactures products ideally suited for integration into critical assays such as Western blotting, immunohistochemistry, immunofluorescence microscopy, ELISA, and flow cytometry. No matter the context, basic research to disease therapy, phosphorylated to methylated and beyond, Rockland embraces the challenge to design, produce, validate, and deliver the best antibodies and life science reagents available in the market today. With Rockland, your solution is complete. BioLegend develops and manufactures highly recognized world-class antibodies and reagents at an outstanding value to customers for biomedical research. Our broad product portfolio includes flow cytometry, cell biology, and biofunctional molecules for research in immunology, neuroscience, cancer, cell biology, stem cells, and more. Our aggressive product development program, accomplished through technology licensing, collaborations, and internal research and development, has produced a product offering of over 17,000 products, which have been collectively cited in over 25,000 peer-reviewed journals. BioLegend also offers a wide range of custom services including assay development, sample testing, and conjugation. BioLegend's reagents are supported by superior customer service and a quality management system dedicated to continuous improvement that is certified for ISO 9001-2008 and ISO 13485-2003. Mettler Toledo is a leading global manufacturer of precision instruments and services for use in laboratories and manufacturing. We offer weighing, analytical, and inspection solutions along our customers' value chain to help them streamline processes, enhance productivity, reach compliance with regulatory requirements, or optimize cost. Discover our high-end products such as industrial scales, Mettler Toledo Rainin LLC pipettes, or process analytics equipment. R&D Systems has established itself as a developer and manufacturer of high-quality proteins and the world leader in immunoassays, as well as a producer of antibodies, arrays, cell selection and detection products, and other cell biology tools. We've been developing and manufacturing research reagents for cancer research for over 30 years. R&D Systems, Novus Biologicals, Tokris Bioscience, and Protein Simple are a part of Biotechne, where we provide researchers with high-quality reagents and instruments to optimize and standardize your workflow. Biotechne's newest acquisition, ACD, and its proprietary RNAscope technology, the first multiplex fluorescent and chromogenic in situ hybridization platform capable of detecting and quantifying RNA biomarkers in situ at a single molecule sensitivity, further expands our capabilities into gene expression analysis. Our sponsors have provided us with some helpful resources related to the study of cancer cell signaling, and we've posted these in our resource list on the left side of the screen. You can access and download these documents at any time during the webinar. And with that, let me introduce our first speaker, Dr. Lynn Marie Postovit. Dr. Postovit is the Alberta Innovates Health Solutions Translational Chair in Cancer, the Salwyn Baldwin Chair in Ovarian Cancer, and the Dr. Anthony Newjame Legacy Oncology Chair, as well as an Associate Professor in the Department of Oncology at the University of Alberta, and an Adjunct Professor in the Department of Anatomy and Cell Biology at Western University. He is also co-director of the Cancer Research Institute of Northern Alberta and a member of WCHRI at the University of Alberta. She is an expert in the area of women's cancers, and her research group strives to find new ways to detect cancers earlier and to treat metastatic disease. They uniquely do this by measuring and targeting factors surrounding cancers. These microenvironmental agents allow cancer cells to adapt to and survive the harshest of treatments. It is through that targeting such entities could diminish the ability of cancer cells to evade therapy and spread. In addition to dozens of academic publications in journals such as Nature Medicine, Nature Reviews Cancer, PNAS, and Cancer Research, Dr. Postovit's work has produced 3 patents, one of which progressed into clinical trials for the treatment of prostate cancer and has led to the development of a startup company. 2 additional patents are currently being developed based on discoveries her team has made since moving to the University of Alberta. Dr. Postovit?
Thank you. So today I'll be talking about cellular adaptations in cancer cells and how this contributes to progression and to disease metastasis and therapy resistance. And we'll be focusing mostly on how the tumor microenvironment and certain features of that may help to promote this by driving cancer stem cell phenotypes. So in this first slide, we're looking at one of the major problems we believe in the treatment of cancer and the treatment of many solid malignancies, such as breast cancer. In these malignancies, we are able to often cure the cancer, particularly if it's at an early stage, by removing the primary tumor and then treating the patient with chemotherapy in either a neoadjuvant or adjuvant setting. And often this works very well, and we're able to cure that patient's cancer past 5 years. However, in certain cases, residual disease remains, or the cancer reemerges later on. In this particular situation, we would have a disease that is more refractory to additional therapies, so more difficult to treat. We would then come in with second-line therapy, and the cancer in some cases may continue to spread and progress, and that cancer will often be even more resistant to therapy. And of course, in the end, it's these cancers that have spread throughout the body through metastases that are chemo-resistant that ultimately will take the patient's life. So in our laboratory, we look at the concept of plasticity, and this is just an analogy that we have here. What you're looking at is a snowshoe hare, and I put this here because it's actually kind of unique to northern places. So we would see this in Edmonton, where the University of Alberta is, and these are fairly large rabbits, and you can see here it's white, and it stays white through the winter here. And these rabbits now, of course, are brown and have an adaptation to change their color during the season so that they can evade being eaten. So we are thinking of this in terms of plasticity. So in the case of the hare or an animal like this would be the ability to change in response to the environment, in order to survive. When we think about cancer, we would think that perhaps this plasticity, so this ability to change and to adapt in response to the microenvironment, and this could be, for example, at the metastatic site, would allow for a selective advantage. And when we think about metastasis, I give the example of breast cancer. So if we were to take a normal breast epithelial cell and take it out of the breast and place it into the lung where breast cancer metastases often occur, that cell would not grow and would not thrive. In fact, it would probably receive the incorrect cues and simply apoptose and die. And of course, this is in contrast to metastatic breast cancers, which can adapt to that environment and thrive. We can also think of plasticity in modern terms, utilizing the cancer stem cell-like theory. And in this particular theory, we would posit that there would be a subset of cells within a tumor that had stem cell-like properties. So this, of course, would be the ability to self-renew and to differentiate. In our laboratory, we see this as a population that's particularly plastic. So a subset of cells that are better able to change in response to the microenvironment to adapt. And of course, this would lead to adaptation to therapy, so resistance to therapy, as well as the ability to metastasize. So what does plasticity look like with the assays that we utilize in our laboratory and when looking at tissue? The first aspect is really a loss of structure function, and this really points to the grade of a tumor. So the loss of normal structure and the acquisition of what looks like an embryonic phenotype or embryonic-like tissue. And this, in fact, was proposed over 150 years ago by Conheim and Durante, and they really looked under a microscope and said that advanced cancers look a lot like embryonic tissue. They lose their normal structure. In addition, we see, particularly for epithelial cancers, processes such as epithelial to mesenchymal transition. So this is the loss of epithelial markers and phenotypes such as E-cadherin at the cell membrane, concomitant with the acquisition of mesenchymal phenotypes such as the expression of vimentin, for example. And so we use this as a tool to measure plasticity in epithelial cancers. This, of course, leads to a functional plasticity. In this, we are looking at survival, self-renewal, and adaptability. So some of the metrics we use are sphere formation, so the ability to form a sphere from a single cell, and that sphere can be broken down, and of course more spheres can be made. This would also be the ability to form tumors from single cells, so limiting dilution assays, and then of course the ability to adapt to harsh environments such as hypoxia, and even to certain stressful treatments such as chemotherapy. And then finally, cancers can also manifest tissue mimicry, where the cells actually can behave like other cell types. And vasculogenic mimicry is one such example where cancer cells can express proteins such as vasculoendothelial cadherin and even partake in vasculogenesis. So how do cancer cells become plastic? In order to conceptualize this, I'm showing here a diagram that was published by Dr. Yamanaka several years ago describing induced pluripotency, and I've overlaid concepts of cancer on top of that. So when you look at the diagram, you can see Waddington's epigenetic landscape, where at the top we have our totipotent cells. Now, during development, these cells sense microenvironmental cues and become increasingly more differentiated, rolling down the hill, so to speak, and into the little crevices at the bottom. These differentiated cells would represent cells such as neurons and skin cells, anything you can think of in the body. Prior to induced pluripotency, it was thought that it would be difficult, if not impossible, to move back up the hill to a pluripotent state, and almost as difficult to transdifferentiate or to overcome these hills into other differentiated states. However, of course, Dr. Yamanaka and colleagues, as well as others, showed that this, in fact, isn't that difficult. We can use our key pluripotency factors, such as NANOG, and move these cells right back up the hill. What I have indicated with the lightning bolts are the genetic mutations that would signify a cancer, because although we believe cancer is an epigenetic disease, it is and always will be also a genetic disease. And I make a note of this because it turns out that many common genetic mutations, such as the loss of p53 or mutations to p53, for example, can actually enhance the reprogramming process. So a loss of p53 can increase the frequency of clones that arise when we're inducing pluripotent stem cells. In addition, the microenvironment of tumors, which include features such as hypoxia, can also help to increase the efficiency of reprogramming. So we're proposing that the genomic alterations of cancer, as well as some of the microenvironmental features, may actually help to allow for epigenetic permissivity. And so the big underlying question in the lab is really, how do cancer cells acquire this plasticity? So what is it about the tumor microenvironment and compounded with some of these mutations that allow these cancer cells to move back up that hill and to acquire some pluripotent or stem cell-like features? So this is the paradigm and the working model that we have in the laboratory. And on the far left, you can see the embryonic stem cells, which would be considered the most pluripotent model we have in our lab, moving down that epigenetic landscape into the well-differentiated tissues of the body. And we're looking now here at advanced cancers on the far right, where we're seeing a case kind of a development undone, or the acquisition of some stem cell-like features. And you can see that there are certain commonalities between these two, such as a reduction in oxygen. I will point out, though, that the stem cell side is extremely regulated, whereas the cancer side is not. So one must always remember when we're studying this that while stem cells are able to self-renew and sense their microenvironment, their job is also to differentiate into tissues. So for every self-renewal factor, there is a factor that would enable or promote differentiation. And we feel that in cancer, this balance is actually lost, and that perhaps understanding this balance and where errors are occurring could be an avenue to help prevent plasticity in cancer. So to address this, now several years ago, while working with Mary Hendricks, we rationalized that cancer cells probably don't use unique signaling pathways, and that they probably hijack normal embryonic or stem cell-like signaling pathways in order to become plastic. And as I've mentioned, in contrast to normal stem cells, these cancer cells would be out of balance, so they would not have both sides of the story, so to speak. So if we were then to put a cancer cell into an early embryonic environment, we'd expect that we would be able to throw that environment off a bit, so to alter development, and that we could look at the alterations and take a step back and then figure out what factors might be being secreted by the cancer cells that could be affecting stem cell fate. So in order to do this and to test this hypothesis, we used a zebrafish model. So you can see here we utilize zebrafish at the high stage, so this would be prior to gastrulation, so an open field, injected fluorescently labeled cancer cells either at the animal pole, as you can see in the far left, or at the margin. And this, by 12 hours, would be expected to affect the fish by altering some development at the head if we're injecting at the pole, or perhaps throughout the entire axis if we were to inject near the margin. What we found when we conducted this assay was interesting. On the far left, you're looking at a cell line called C81-61. This is a melanoma cell line that is non-tumorigenic and very well differentiated. The center is a vehicle control. And the far right is a C81-61. This came from the same patient as C81-61, except that it's from a metastatic lesion and is highly dedifferentiated. In this particular experiment, we injected the cancer cells at the animal pole and then looked to see if there were effects on the head of the zebrafish. As you can see, the well-differentiated cancer cells really didn't do anything. However, the poorly differentiated cancer cells induced a bump on the head of the zebrafish. So our initial thought was that perhaps this was simply a tumor on the head of the fish. So we decided to look at what the bump, for lack of a better description, was made of. To do this, we utilized whole mount staining. What you're seeing here is beta-catenin. This is fish beta-catenin in red, and then the cancer cells are fluorescently labeled in green. And you can see immediately that the cancer cells are not forming a tumor. Rather, they are organizing the formation of this structure by the fish cells. So something the cancer cells are secreting and putting out was inducing this structure. Now, when we did the experiment by injecting cancer cells into the margin, so this will affect the entire axis, and then waited a couple of days, what we saw was a duplicated axis formation. So on the far left, you have a fish with essentially two heads, 1, 2, 3, 4 eyes, with the cancer cells at the apex, and the cancer cells are just in the fluorescent diagram and outlined by green. So these cancer cells were inducing an ectopic or secondary axis, and it turns out that few things could do this so thoroughly, and we did a number of experiments to eventually come down to a factor called Nodal. So Nodal is an embryonic morphogen belonging to the transforming growth factor beta superfamily. So it's going to be secreted, and it's going to work as a homodimer in the extracellular space. In development, Nodal initiates embryonic axis formation, so that development of an axis as we saw in the fish, as well as left-right asymmetry. So Nodal is restricted to the right side of an embryo by its inhibitor, lefty, and this is very important for left-rightedness. We found that Nodal is expressed in poorly differentiated cancer cells like C8161, but not in well-differentiated counterparts like C81-61. And this is held true and has been shown by us and by groups around the world in cancers such as breast cancer, pancreatic cancer, prostate cancer, brain cancer, and the list goes on. Importantly, Nodal signals to affect the epigenetic landscape. So as it signals, it works to combine with epigenetic factors, leading to a reduction in H3K27 methylation at certain areas, as well as the acquisition of H3K4 methylation at certain areas. So essentially, it's helping to work at bivalent promoters to regulate gene expression, and this can be through the genome. Nodal is also required for the maintenance of an undifferentiated state in embryonic stem cells. So it's a very potent factor that both induces differentiation in cases and then also represses it in some cases. Now, when we look at cancer, we see that nodal expression correlates with that loss of structure function that I mentioned previously. The top panel, we're looking at melanoma. The bottom panel, we're looking at breast cancer. And we're going from normal tissue to the least differentiated metastatic tissue. And what you can see is that nodal expression in melanoma begins to arise at the vertical growth phase when there's a loss of structure function, and then is present in the vast majority of metastatic cancers. When we look at breast cancer, we see that similarly, nodal expression is not in the normal breast and is actually very rare in stage 1 disease, but then begins to emerge in stage 2 and beyond. So these invasive ductal carcinomas and then in metastatic cancers. And a study done a few years ago showed that nodal actually correlates with stage, so this conversion from stage 1 to 2, 3, and beyond, but not ER, PR, or HER2 status, and that it's expressed in over 85% of invasive cancers, so stage 2 and above, irrespective of the subtype. And so this is something that really is correlating with that loss of structure function. Now, we found nodal originally in melanoma and have since looked at breast cancer, but others around the world have shown that nodal similarly is correlated with poor progression, free survival, as well as the acquisition of stem cell-like phenotypes in a variety of cancers listed here. So prostate, testicular, endometrial, brain, pancreatic, hepatic carcinoma, and the list really goes on. So what is it that nodal does. And the answer to this question is it seems that it does quite a lot in a very context-dependent manner, which when we consider its function in development probably isn't so surprising. I'm going to show in this next slide an example of how much Nodal can actually do. So in this particular experiment, we're taking MB231 breast cancer cells. These are triple negative claudin low breast cancer cells that are poorly differentiated and express Nodal. We are then subjecting these to SILAC-based proteomics. So we're going to knock down Nodal in one of the cell lines and then allow it to persist in the other one, label one but not the other, and then combine them and perform mass spec. And in this experiment, we're performing mass spec specifically and only on the secretome. And what I want to highlight are the number of proteins just in the secretome that change in response to the alteration of Nodal. And this isn't, as I said, incredibly surprising, as Nodal we expect to go through the genome, and in addition to transcriptionally regulating the expression of genes, also epigenetically regulate them. And you can see here that in the case of this breast cancer model, we're actually modulating a number of cytokines, including IL-6. So these are signified across the top. C is simply signifying the volcano plot, showing the differences between nodal expressing and nodal non-expressing cells, and then conformation with an ELISA is on the far right bottom corner. So what exactly is Nodal doing and how is it doing it? So before I get into this, I'm just going to briefly describe nodal signaling. As I mentioned previously, Nodal is a homodimer. It's a TGF-beta family member. It's secreted into the extracellular space, and the canonical signaling pathway has it signaled through activin-like kinase 4/7 as well as ACT2, our 2B, so very canonical TGF-beta signaling, and this is enhanced by the co-receptor crypto, a GPI-linked co-receptor. Activation of the receptor complex leads to phosphorylation of SMAD2 and perhaps 3, which then can work with SMAD4 to regulate transcription. This can also work to associate with factors such as Jumanji proteins to affect epigenetic marks throughout the genome. And I'll add that Nodal can also induce the MAP kinase ERK1/2 signaling pathway as well. So the first question that we're going to address here is whether perhaps Nodal can directly enhance these stem cell-like phenotypes. And I'm just showing this figure again as a reminder of some of the assays that we use to test that. So we look to see whether there's an increase in self-renewal by doing sphere formation assays as well as limiting dilution assays or tumor formation assays in vivo. We also look for the acquisition of stem cell-like phenotypes and gene expression patterns, including epithelial to mesenchymal transition. So in this set of experiments here, we're looking at breast cancer cells. At the top left, we're taking a T47D breast cancer cell line. This particular cell line is a luminal B. It's fairly well differentiated and poorly metastatic. And because Nodal is a secreted factor in this particular model, we're simply adding recombinant Nodal into the niche and then conducting sphere formation assays. And you can see that as we add more Nodal, we increase the number of spheres that are formed. On the bottom, we have MB231 cells, which express Nodal. We see that we have a reduction in sphere formation when Nodal is inhibited, and this can be rescued by recombinant Nodal. And then on the far right, we're simply inhibiting the Nodal signaling pathway with an inhibitor and showing a reduction in both the number and the size of the spheres that are formed. Here is just an extension of that, looking at the accumulation of Nodal in spheres as compared to monolayer, with the idea that we're going to have an enhancement or an enrichment of cancer stem cells within the spheres. And we show that this is in fact the case whether we make spheres in a bioreactor or on low attachment plates. Nodal levels are increased, and this of course is blocked if we knock down Nodal expression. This also holds true for markers of breast cancer stem cells. So in breast cancer, this population is thought to be enriched by a CD44 positive, CD24 negative subpopulation, and we see that this is mitigated when we start to inhibit nodal. We have looked also, as I mentioned, at the EMT phenotype. So in this particular case, we're using these 247D cells again, but with a genetic alteration where we have an overexpression of nodal. In this particular model, the overexpression of nodal results in an increase in the expression of mesenchymal proteins such as vimentin, as well as transcription factors such as twist, and this is concomitant with the silencing of the estrogen receptor. At the bottom, we're looking at E-cadherin, and we can see that while nodal expression does not reduce the levels of E-cadherin per se, it does cause a translocation of the protein from the membrane into the cytoplasm, so suggesting a general EMT effect in response to nodal, but one that isn't exactly like TGF-beta, which also induces a loss of E-cadherin. So we have some differences that we're observing here. Of course, probably not surprisingly, the alterations in EMT are associated with alterations in cellular invasion. So here we're looking at MB231 cells. We're implementing an assay where we take spheres and place them in collagen. In this particular assay, we're utilizing a blocking antibody to nodal and demonstrating that this blocking antibody inhibits both the number of cells that invade as well as the distance that they're able to invade. So we next wanted to understand whether nodal would affect stem cell-like phenotypes in vivo. So to do this, we're going to use this NOD-SCID mouse, injecting through the tail vein. In this particular model, we wanted to dissect the ability to seed from the ability to grow. So we want to know how many of those cells that seed are able to form a colony, because we felt that would be more indicative of self-renewal. So to do this, we're going through the tail vein. We're looking at 4 weeks to see if seeding is altered, and then 8 weeks to see if growth is altered. Doing this, we determined that at the 4-week time point, there really isn't a difference in the seeding of the cells. You can see here, what you're seeing is actually staining for GUS-B. This is on the background of a GUS-B knockout mouse. So we can actually get down to the single cell level by simply using an enzymatic reaction. So we're able here to tell that, yes, our seeding is pretty consistent between the two. However, if we look out to 8 weeks, we see a dramatic difference where the nodal knockdown cells are unable to form macrometastatic lesions in this particular cancer. So how about metastasis and in a more kind of clinically relevant model? So to address this, we used an inducible system. So in this particular model, we have an orthotopic injection of the cancer cells into the mammary fat bed, in this case of NSG mice, to enable metastases. We can then treat with DOX in order to reduce nodal expression in a certain subset of cells. So we inject the mice, we allow them to grow, and then we start feeding them DOX, and then look to see whether certain elements of tumor formation and metastases are altered. So when we do this particular model and look at the lungs, we actually didn't see any difference between the different treatment types. And we don't know if this may be because we initiated DOX treatment following seeding, or if in this model the lung is still permissive. However, when we look at the liver, we saw dramatic differences in our treatment types. So we see that the control animals have metastases to the liver, as do the SH nodal animals that have not been treated with DOX, so that would be expected to have nodal. However, we see very few lesions in the animals wherein nodal was downregulated, and this is just quantified here. So in summary, we believe that cancer cells are a bit like a recapitulation of normal stem cells, and that they do this by hijacking certain developmental pathways. And we think that perhaps nodal may be one such pathway, and that targeting nodal either with inhibitors or by understanding endogenous modes of regulation may be a therapeutic strategy to be used along with chemotherapy, as an example, to prevent adaptation and perhaps even metastases. So the next question that arises when you look at the pattern of nodal expression is really, how does nodal get turned on in cancers? So how do these cancers obtain plasticity? And we've done some genomic analyses and shown that this isn't due to an amplification of the gene. It's really epigenetic and likely driven by the microenvironment. So the first microenvironmental factor we thought to look at was hypoxia. And this is simply, in the case of breast cancer, due to the fact that hypoxia occurs just as a simple result of rapid tumor growth. So in this particular diagram here, you see what would be considered the breast duct with the basement membrane and the blood vessels on the outside of the basement membrane. Now, as the cancer forms and grows, they're going to fill the duct. And as you can imagine, the cells in the center would be far away from the blood vessels, and so you'd have increasing hypoxia as the tumor grows. And so we hypothesized that perhaps this hypoxic environment might help to drive the acquisition of nodal and some of these stem cell-associated phenotypes. And this isn't really that far of a stretch, as hypoxia has been shown to promote induced pluripotency, to help sustain stem cell phenotypes in a number of normal stem cells, and also to upregulate cancer stem cells in a number of different cancer cell types. So the first thing that we did was simply take our breast cancer cells and expose them to decreasing levels of oxygen. That's what you see on the top left here. We're looking at nodal that would be in the conditioned medium. This is cleaved and active nodal, and then nodal in the lysate, both the pronodal as well as the active nodal at 15 kilodaltons here. And the main take-home message is that yes, low oxygen levels increase nodal expression, and that this occurs over a period of 24 hours, and it occurs in breast cancer cells, but then also in normal stem cells, the H9 human embryonic stem cells seen on the right. We can also see that this is not exclusive to in vitro culture. In this particular model here, we're actually using a xenograft of some 149 breast cancer cells, pulling out the hypoxic fraction with CA9 and then measuring nodal. And you can see that with the histogram here that nodal expression is highest when CA9 expression is also high. We can also see this in patient-derived xenografts. These are 3 different patient-derived xenografts with very different levels of hypoxia. Hypoxia in this case is measured with CA9, as seen in the bottom panel, and nodal expression is in the top panel. And what we notice is that nodal co-localizes very well with hypoxia, even in these patient-derived xenografts, and this also occurs in patient tissues. How about these stem cell phenotypes? Well, here we're taking MB231 cells that already have a high number of cancer stem cells, and in confirmation of studies published in the literature, exposure to 1% oxygen or hypoxia increases the number of cancer stem cells, and this can be blocked if we inhibit the TGF-beta and nodal signaling pathways. So from a signaling perspective, how is this occurring? Of course, when we think of hypoxia, the first thing we're going to look at is hypoxia-inducible factor. So hypoxia-inducible factor alpha is a transcription factor that under normoxic conditions is hydroxylated and then sent to the proteasome for degradation. However, under hypoxic conditions, the hydroxylation no longer occurs, HIP is stabilized and able to regulate the transcription of a number of different genes. So we looked at the regulatory region of nodal to see whether we could find classical hypoxia responsive elements. And it turns out that really there were none in that particular region. But there is in the nodal regulatory region one enhancer that is responsive to Notch, so the NDE that's shown here on the left in green. And there's a link between Notch and HIF signaling. So as you can see here, it's been shown that Notch, when bound to its ligand on an adjacent cell and is active, can actually work with HIF such that it stabilizes HIF and then moves to Notch-responsive elements, as you can see here in the diagram, to help induce expression. So we thought, let's see if perhaps HIF is working to induce nodal through this Notch-dependent mechanism. So the first thing we did was simply to modify HIF expression. And I mentioned HIF1, but there's also two other isoforms, HIF2 and HIF3. HIF2 also regulating the expression of hypoxia-induced genes. So in this particular case, we're using siRNA to either HIF1 or HIF2, and then looking at nodal expression. So you look across the top, you see that nodal is induced by hypoxia. However, if we knock down HIF1 alpha, nothing occurs. If we knock down HIF2 alpha, we still have an upregulation. So what we're showing here really is that HIF1 alpha seems to be required for the hypoxic upregulation of nodal in this particular cell type. We're now looking at just a simple overexpression of a HIF1 alpha and HIF2 alpha, and this is really confirming what we've seen before. So the idea is that if HIF1 alpha is truly helping to increase nodal expression, we should be able to induce nodal with HIF1 alpha overexpression regardless of the oxygen concentration. And that's what we're seeing here. So the fluorescent diagrams are showing that our transfections work, we get translocation to the nucleus, and the westerns at the bottom simply show that when we induce HIF1 alpha but not HIF2 alpha, we upregulate nodal. So in terms of this regulatory region, we wanted to understand whether HIF was acting through the NDE. So we use a promoter assay, as you can see here. In this particular assay, we have a wild type. We have one where we have one of the sites mutated. There are two notch-dependent sites in this area, so either one of them or both of them together. So we're going to use this reporter assay to see whether hypoxia induces activity from this enhancer region, and also whether HIF does. And the take-home message is that this is in fact the case. So across the top you see that hypoxia increases the reporter, as does HIF1. HIF2 does not. And the induction by both hypoxia as well as HIF1 can be mitigated if we remove either one of the notch binding sites. So when we look at the nodal expression in our tissues, we can also see that it's quite robust and that it's continually there. And so we started to think that maybe something else was at play. So even when you have reoxygenation, you have nodal expression. And it turns out that nodal works via a positive feedback loop. And so we started to ask the question whether perhaps nodal is maintained in these cancer cells by this positive feedback loop. So nodal would induce its own expression and then signal back to itself and then enable the continuance of that expression. So here we're testing this hypothesis. So the idea is that if this were to be the case, then if we were to inhibit the receptor, we would block the ability of nodal to maintain this positive feedback loop. And that's what you're seeing here. So here's nodal upregulating, hypoxia upregulating nodal expression in the first two of this Western blot. As we move through, we have hypoxia reoxygenation. So this is nodal being maintained at 24 hours and that being blocked if we inhibit the receptor. And then this also occurs at 48 hours. And you can see at the bottom just immunofluorescence of SMAD2 in the nucleus, demonstrating that it is probably in fact this signaling mechanism that's helping to maintain nodal expression. So what we think is happening here is that we have nodal or hypoxia and combining with notch and going to the notch responsive element in the nodal regulatory region. This we think leads to the production of nodal, which will then be secreted into the extracellular space where it will initiate nodal signaling. This will subsequently allow nodal to upregulate itself conventionally through transcription, and we've also shown likely through epigenetics as well. So even once hypoxia is taken away, nodal can maintain itself via this positive feedback loop, as we can see here. And I seem to have a duplication of slides, so we'll just move through these. Okay. I apologize. So in conclusion, we see cancer in the acquisition of plasticity on an epigenetic platform. And we believe that we can understand this acquisition of plasticity by looking at embryological models and understanding cancer is, yes, a disease of development undone, but one that is dysregulated. And so we propose that coming in and inhibiting factors that maintain plasticity, like nodal, perhaps WNTs and others, could be used as a way to help inhibit adaptation, prevent metastases, and also disease recurrence. So with that, I just want to thank our funding organizations that have supported our research through the years, as well as key members of the team who've done this work, including Guiwa Zhang and Daniela Quayle, Megan Taylor, Michael Jewer, amongst many, many others. And thank you for your attention.
Thank you, Dr. Postevit. As a reminder to our audience, you may ask questions at any time by typing your query directly into the question box. The panelists will have a chance to respond to your questions at the end of their presentation. Our second speaker today is Dr. William Weiss. The Weiss Lab has developed mouse models for tumors of the nervous system, including genetically engineered mouse models for glioma, medulloblastoma, and neuroblastoma, as well as more novel models for these tumors based on differentiation and transformation of human iPS cells. Dr. Weiss's lab has established these as platforms for biology, genetics, and developmental therapeutics, having published preclinical trials using both chemotherapeutic and targeted agents in parallel with analyses of primary human tumors. Their focus is on EGFR, PI3K, mTOR biology, chemical genetic approaches to kinase function, and developing inhibitors of kinases that stabilize MYC family proteins as a MYC-directed therapy. Dr. Weiss co-leads the Pediatric Malignancies Program within UCSF's Helen Diller Family Comprehensive Cancer Center. He is a senior editor of Cancer Research and an associate editor of Molecular and Cell Biology, Scientific Reports, and Neuro-Oncology. He received a BS from Tufts, an MD-PhD from Stanford, clinical training in pediatrics and in neurology at Boston Children's Hospital and UCSF, and was a postdoc with Mike Bishop. Dr. Weiss?
Hi, thanks very much. So I'm going to shift a little bit and start talking about targeting a protein called 4-ABP1 in glioblastoma. So adult glioma is the most common primary brain tumor, and this can be divided into astrocytic and oligodendroglial lineages. The astrocytic lineage is probably 95% of tumors. And the tumors can be graded, grades 2, 3, and 4. A vast majority of patients present with highest grade 4 disease, and a minority, probably 5 to 10%, progress from a low to a high grade intermediate. And so just because people often think about stage rather than grade, I give an example here of two tumors that differ in stage. There's a big tumor on the left and a small tumor on the right, but are the same in terms of grade. And so these two patients have the same prognosis, despite the fact that one has a huge tumor burden and the other one doesn't. The histology is very characteristic and includes, on the left side, pseudopalisading necrosis. So the center of the left slide shows a necrotic area, and the cells that are lined up sort of in a column around it are pseudopalisading. The right histology slide shows anaplasia. So the cells you can see there are quite non-uniform in appearance. Glioblastoma used to be called glioblastoma multiforme, and the multiforme moniker is one I personally liked and was sad to see that it's no longer in place. But I think the slide on the right shows the multiforme nature of this disease. And the outcome for this tumor is fairly dismal. So again, those two patients had the same disease, and their prognosis is pretty much the same. So this shows you the standard of care since 2005, when the last real big paper came out, and that shows that with radiotherapy, after surgery, with radiotherapy, the patients lived an average of about 12 months. And when you added an alkylating agent called temozolomide, you move that curve to about 18 months. So, that is no small feat when you're have this tumor and you may die in a year, you'd probably welcome the 3 months extra, but it's clear that we have a long way to go. So glioblastoma has also been the poster child for the Cancer Genome Anatomy Atlas. And my slide is not showing on here. There we go. And so the TCGA has identified or re-identified 3 major pathways that drive these tumors. Each of these is mutated in about 80% of patients, so there's a lot of overlap among the pathways. And these can be categorized in a number of ways, but for purposes of today's argument, we'll use this categorization, which is RTK signaling, P53 pathway signaling, and RB pathway signaling. And I'm going to focus today on RTK, receptor tyrosine kinase signaling. So in my own redrawing of that same cartoon, I show the growth factor receptors at the top middle of the slide signaling to PI3 kinase. On the right side would be nutrients and that would be nutrient signaling. So there's this coordinated signal from growth factors and nutrients through PI3 kinase, AKT, and mTOR. And mTOR is a major regulator of protein synthesis. So mTOR signals through 2 outputs we'll talk about in a minute, but the one I show here is called mTORC1, and the 2 outputs are P70S6 kinase, which regulates ribosomal biogenesis, and also 4-ABP1, which signals to the elongation initiation factor EIF4E to regulate cap-dependent translation. On the left side of the slide, you see that EIF4E is normally inhibited by 4-ABP1, and when mTOR phosphorylates 4-ABP, EIF4E is now active and can regulate cap-dependent translation. So mTOR signals to make protein, and it signals to integrate nutrient and growth factor signaling to regulate protein synthesis. Protein synthesis is the most energetically costly decision a cell makes. So when there's abundant nutrients and growth factors in the bloodstream, mTOR turns on protein synthesis, and when those growth factors and nutrients are limiting, mTOR turns off protein synthesis. It's perhaps not surprising then on the next slide that we see that mTOR is actually also a major regulator of hunger and satiety in the hypothalamus. So when your nutrients and growth factors are in low supply, mTOR signaling is what tells you to eat so that you can bring them to higher supply, and it regulates nutrient. It regulates the intake of nutrients and is signaled too by levels of insulin, leptin, and nutrients, and it regulates hunger. Okay. So again, as I mentioned earlier, mTOR actually exists in 2 protein complexes. One is called TORC1, and the other is called TORC2. And these, it's the same kinase, but it is actually in complex with a number of different proteins. Both mTORC1 and mTORC2 are downstream of AKT. mTORC2 is also upstream of AKT. So whereas AKT regulates TORC2, TORC2 also phosphorylates and regulates AKT in a rather unusual arrangement. But for today's talk, we're going to talk about TORC1. And so if you look at this pathway in overview, the link between growth factor receptors to PI3 kinase, AKT, and TOR leads a lot of potential targeting approaches. And in clinic, we have receptor tyrosine kinase inhibitors in clinical use, PI3 kinase inhibitors in clinical use, and mTOR inhibitors in clinical use. I think AKT inhibitors are in clinical development. So how do we best target this axis in glioblastoma brain tumors? So what we've shown, what we and others have shown in prior work is that the receptor tyrosine kinases and the PI3 kinases signal to AKT, and you can block those and get an anti-proliferative response only if the signaling axis linking this pathway together is intact. But that pathway is frequently not intact in glioblastoma. PI3 kinase itself can have gain-of-function mutations, and P10, which is a negative regulator of PI3 kinase, has loss-of-function mutations. And collectively, this means that if you give an inhibitor that acts upstream of these gain-of-function mutations, you don't affect downstream signaling. So the receptor tyrosine kinase and PI3 kinase inhibitors fail in glioblastoma due to this mutational activation of PI3 kinase signaling. So what about AKT inhibitors? So another issue that we have demonstrated in the past is that even though there's this canonical signaling of PI3 kinase to AKT to TOR, in glioblastoma this appears to be rewired, and we have not seen glioblastoma cell lines in which blockade of PI3 kinase or AKT actually signals downstream to negatively regulate TOR. We certainly see this in many other breast cancer lines, so I don't think it's operator error in our hands. It's just rewiring. And so this phenomenon is illustrated in this recent paper that we published, and that will be the focus of today's discussion. And this is work by Chi-Wen Fan, who's a faculty member in my laboratory. And so what this shows, between the red bars on the left, you see this drug called INC1437, which is a PI3 kinase alpha inhibitor, and it's being dosed into a glioblastoma cell line at 100 nanomolar, 1 micromolar, and 10 micromolar. And on the top half of the slide shows cell cycle regulation, and the bottom half shows biochemistry. And what you can see is on the top half, you get a very subtle G1 arrest. The blue bars get bigger. And in the biochemistry, this correlates with the blockade of AKT, which shows dose-dependent decrease in phosphorylation of AKT. But the phosphorylation of RPS6, a TORC1 target, and of 4-EBP, a second TORC1 target, is unaffected. Similarly, an AKT1/2 inhibitor, shown on the right side between the two red bars, shows a very similar result with very modest G1 arrest, dose-dependent decrease in phosphorylation of AKT, and no real effect on the phosphorylation of RPS6 or of phospho-4-EBP1. So again, this argues that there's a miswiring of signaling between AKT and TOR in glioblastoma, such that blockade of PI3 kinase and AKT fails to translate to blockade of mTOR. And I guess I'm sorry, I meant to march through that with some blocks, which I forgot I put in, so I'll skip ahead. Okay, so what about targeting mTOR directly, if mTOR seems to be a critical target for efficacy? So the first generation of mTOR inhibitors include rapamycin and derivatives of rapamycin, which can be called rapalogs. That's shown by the circled R here. And these drugs bind to abundant cellular protein called FKBP12. The complex of rapamycin and FKBP12 binds to part of TOR called the FRB. And this changes the ATP and substrate binding pocket allosterically so that it actually blocks access of a single substrate, S6 kinase, but not of 4-EBP1. And because of the location of FRB in the TORC2 complex, these drugs don't work very well against TORC2. So they're TORC1 specific, but within TORC1 they're selective for S6 and not for 4-EBP. And that's shown in this slide. If you look at rapamycin, if you look at rapamycin, you can see that it doesn't show much of an anti-proliferative effect. We get a feedback activation of AKT. This has been well described activation. And you get blockade of phospho-RPS6 in a dose-dependent manner, but you don't get blockade of phospho-4-EBP. So rapamycin blocks phospho-S6, but not phospho-4-EBP. And it turns out that phospho-4-EBP is a better biomarker for efficacy. And that you could probably see that to the very right of the rapamycin, this mTOR kinase inhibitor, which shows that we can block AKT in a dose-dependent manner, block phospho-RPS6 in a dose-dependent manner, block phospho-4-EBP1 in a dose-dependent manner, and in this case we can block proliferation with a very strong G1 arrest in a dose-dependent manner. So this shows the way these TOR kinase inhibitors work in this next slide. So this is an orthosteric kinase inhibitor. It actually blocks ATP and substrate access in the TOR kinase pocket, and as such, it fully blocks TORC1 and TORC2. The downstream functions are S6, 4-EBP, and AKT, as I showed you in the previous slide. And so I showed you the result of the TOR kinase inhibitor. If we actually look at GDC941, which is a mTOR which is, I'm sorry, a PI3 kinase inhibitor from Genentech that has about 10-fold selectivity for PI3 kinase over mTOR, you can see that we block phospho-AKT even at the lowest dose, and we're fully blocked at the 1 and 10 micromolar dose. We block phospho-RPS6 in a dose-dependent manner, and we block phospho-4BP1 in a dose-dependent manner. And so basically at the lowest dose, which is 100 nanomolar, we're not blocking mTOR, but at the intermediate and highest dose, we are effectively blocking mTOR. And the anti-proliferative effect is clearly correlating with the blockade of 40BP1 and of mTOR more than it's correlating with the blockade of PI3 kinase. On the very right side of the slide, it was a Novartis tool compound called BEZ235, which blocks both PI3 kinase and mTOR. And you can see that even at the lowest dose, we have a strong anti-proliferative response, G0, G1 arrest in the blue bars. We have fully blocked AKT. We have a pretty good effect on RPS6, which doesn't get much better. And we fully blocked 40BP1. Phospho-40BP1 is gone. And so, again, the orthosteric TOR kinase inhibitors and PI3 kinase TOR kinase inhibitors effectively block proliferation, and that proliferation block correlates with blockade of phospho-40BP1. I think it's interesting, just when we talked about feedback, we also did, we looked at levels of PIP3 in response to rapamycin and the TOR kinase inhibitor KU63794. And what you can see, which I showed you previously, is that rapamycin activates AKT, whereas the Kudos compound, I show you here, does not. If you actually look at PIP3 levels, you're getting feedback activation in both cases. But since rapamycin doesn't block AKT, you can see AKT go up. The TOR kinase inhibitors are blocking TORC2, so that is blocking AKT. So AKT is not going up in response to PIP3. But we are getting a nice dose-dependent blockade of RPS6 with both drugs, but we only get a blockade of 40BP with the Kudos compound, the KU drug, and not with the 40BP drug. Okay, so I think what was surprising, given that I showed you that TOR kinase inhibitors are much better at blocking 40BP than the other drugs I showed you, we put these head-to-head in an orthotopic glioblastoma model in vivo. So we delivered these drugs systemically to mice that had brain tumors. And what we showed was actually that MLN128, which is now called TAC228, which is one of the lead clinical TOR kinase inhibitors, does not outperform rapamycin. And so the green curve shows rapamycin, which gives a modest improvement in survival. The MLN128 is the red curve, which is intermediate between vehicle and rapamycin. So if we trace this to biochemistry, we can see why. So these are 3 different tumor samples treated with vehicle MLN128 or rapamycin. And if we zoom in on the phospho-40BP1 lane, you can see that rapamycin and the MLN128 are equivalent in blocking 40BP, maybe even rapamycin's a little better. And so this is surprising at 2 levels. One, that rapamycin has in vivo efficacy, whereas it had no in vitro efficacy. I don't really have an insight into that. But the other is that MLN128 is so modestly effective in vivo when it was so highly effective in vitro, which I will get to. So where do we go next? We really want a durable 40BP1 inhibitor. So rapalogs get into the brain but have shown limited activity in glioblastoma. Second-generation TOR kinase inhibitors get into the brain but also show poor activity. And so with my collaborator, Keyvan Shokat, we tested a third-generation TOR inhibitor that he generated, hypothesizing that the rapamycin backbone, which I'll show you in a minute, would help these drugs get into the brain, and improved FKBP12 binding would improve the pharmacodynamics. And so I want to remind you again in this crystal structure by Baratek and Williams that rapamycin, shown in yellow, you can see it, is binding to this orange protein called FKBP12, and the 2 of them together bind the FRB, which is a domain of TOR itself, of mTOR itself. And so what Keyvan did was actually link rapamycin to the TOR kinase inhibitor MLN128. And so this drug, which he calls Rapalink, utilizes the protein-protein binding of RAPA for strong affinity to TORC1, and it maintains MLN128, the TOR kinase inhibitor, in close proximity to its target. And so the right top structure is rapamycin, the left structure is RapaLink, and between the parentheses is a linker that links them together. I'm sorry, top right is rapamycin, left side is MLN128, and the parentheses with the N just shows the linker that links them together. So it's one molecule derived from two parent molecules. And so we looked at this molecule in cell culture, and the rapamycin is shown in blue. You can see at sub nanomolar, 0.39 nanomolar concentration, we achieve maximal growth inhibition, which is about 30% growth inhibition. And going up on dose doesn't do much. In contrast, RapaLink, we have to go a little higher to 1.5 nanomolar, but we get really profound growth inhibition to 75%. And that above 1.5 nanomolar, again, we don't do much. And MLN128 in red really gives a dose-dependent decrease in growth. So we have to really get to 200 nanomolar before we see bioequivalence to RapaLink. And if we look at the biochemistry, what you can see in a dose response where the RapaLink is at lower doses than the other two drugs. If we look at 128 first, on the left side, you can see dose-dependent blockade of the TORC2 target AKT. And if you go down, you get dose-dependent blockade of the TORC2 target phospho-NDRG1. We get dose-dependent blockade of RPS6, and we get dose-dependent blockade of 4-EBP. And we see this difference between the dose required to block S6 and the dose required to block 4-EBP. With MLN128, it always takes a little more to block phospho-4-EBP. With RapaLink, we see a real cliff at 1.5 nanomolar. We block both of those targets, RPS6 and 4-EBP. And the AKT blockade, there's a little bit of a differential, but it's not a lot. We get to 6 nanomolar, we're blocking AKT. Rapamycin doesn't block the TORC2 targets, and it blocks S6 really efficiently at very low dose, but it doesn't block 4-EBP. And I think I talked about that. So what about inhibition of TORC2? So for RapaLink, the TORC2 specificity, I think I showed you, was a little less than the TORC1 specificity, whereas for MLN128, as you see in this slide, we're really blocking TORC1 and TORC2 equivalently. So if we look at the distinction here, again, at 1.5 nanomolar, we've completely blocked TORC1 targets, and it's not until 6 nanomolar that we've completely blocked the TORC2 targets. And now if we correlate that with the proliferative block, you see that at 1.5 nanomolar, we've achieved our maximal proliferative block, and as we dial in TORC2 blockade, we're not dialing in more anti-proliferative activity. So this would argue that TORC2 blockade is not important. We've also, in this slide, actually looked at adding an AKT inhibitor to block TORC2 to RapaLink, rapamycin, or MLN128. And so if you look at the first 4 red bars, the first red bar shows our vehicle treatment relative growth. Second red bar shows that MLN128 by itself brings the growth down significantly in this experiment. The third one is RapaLink, and the last one, which is about 30% or 40% blockade, is rapamycin. So this is with these drugs as monotherapy. And if now we add the AKT inhibitor, you can see it has a little bit of activity by itself. It does not augment the activity of MLN128 or RapaLink, but it does augment the activity of rapamycin. And the biochemistry follows that that the AKT inhibitor blocks AKT, but for purposes of time, I probably won't belabor the biochemistry. So this again argues that inhibition of mTORC2 is not really important for the activity of RapaLink. So we next did some experiments to show that RapaLink crosses the blood-brain barrier. We took normal mice and dosed them with insulin to activate the signaling pathway. And in the setting of insulin treatment, we dosed them with MLN128, RapaLink at 2 different doses, or rapamycin. And if you look at the orange boxes there for skeletal muscle, you can see, as compared to the second lane, which is the insulin alone, that that Rapamycin in skeletal muscle gives a very modest blockade at our lower dose, but a significant blockade at our higher dose. And that if we look in the brain, we see a similar level of blockade, perhaps a little bit muted, but clearly the drug is getting into the brain. So I think the blood-brain barrier is a barrier, but not keeping the drug out. We did additional experiments I won't show you where dosing with Rapamycin didn't change weight, liver function tests, or the blood counts over a few weeks of therapy. And so then we moved this into orthotopic xenografts. This is a cell line-derived xenograft, and this is luciferase imaging. And what you can see at the bottom is that Rapamycin takes an established tumor and actually drops it down to it decreases its signal intensity from luciferase by 20 days, although it starts coming back at 25 days. And then the MLN128 is a little less active than Rapamycin, as I showed you before. If we look at tumor burden, you can see that the tumor burden at 10 days with Rapamycin has dropped, and then it starts to pick up again by 25 days. So it's still below baseline by 25 days. And with the other two drugs, the red and orange curve, there's perhaps a little drop by 5 days, but then they pick up steadily, and they're keeping up pretty close with the vehicle control. If we look at in vivo efficacy now, I showed you this slide for MLN128 that we get partial blockade of 4-ABP, and for Rapamycin where we get also partial blockade. But if you look at the Rapamycin with the orange box at the bottom of the slide, you can see it's much better than both drugs. And if we look at the phospho-AKT blot, we see we're not really appreciably hitting the this TORC2 target in vivo. This might be because just not enough drug gets into the brain, but it might be that there's a real distinction between in vitro and in vivo pharmacology. And now if we look at survival, what you can see is that Rapamycin really does much better than the other two drugs. So the green and red curves I showed you before, and you get by about 60 days essentially, or maybe 55 days, all the mice are dead that are treated with vehicle 128 or Rapamycin, and all the mice are alive treated with Rapamycin, but by about 60 days you start losing those mice. And we looked at proliferation, and there's a clear, strong anti-proliferative response, stronger for Rapamycin than with Rapamycin, and this is evidenced in the histology. The H&E is shown on top, the Ki67, which is our proliferative marker, is shown in the brown staining. We've repeated this result and got a qualitatively similar result in a orthotopic PDX patient-derived xenograft, which people feel is better representative of human tumors than cell line-based models. And we basically get the same qualitative result. We block 4-ABP better using Rapamycin compared to the other drugs, and we're not seeing TORC2 blockade in vivo. And we've also done an experiment in a mouse model of medulloblastoma that my lab developed, work from Özlem Aksoy in my lab, where Rapamycin, shown by these luciferase signaling at days 0, 5, 9, 15, 20, and 26, reduced tumor burden much better than vehicle control. And this was mainly done to satisfy reviewers who wanted a blood-brain barrier intact animal where we hadn't manipulated barrier by introducing a tumor through injection. And when we add the biochemistry here, you can see that in vivo we're hitting RPS6, and I should have put the bar around 4-ABP, which is you can see on this slide. And the AKT target, AKT473, is not affected. So what's the basis for this efficacy? We did washout experiments, and we showed that that if we wash out drug and then follow recovery of proliferation at 0, 1, 2, and 4 days, we're looking here at 4 days, you can see that Rapamycin has a much more profound effect than the other two drugs. And if we actually graph this out, you can see that we start recovering proliferation even at 1 day after washing out 128. We don't start recovering until 4 days after washing out of Rapamycin. And Rapamycin is even more durable, but it's a less potent anti-proliferative. If we now do biochemistry, you can see that with MLN128, when we look now in hours rather than days, when we wash out MLN128, from 0 to 1 hour, you see almost full recovery of signaling of phospho-AKT, phospho-RPS6, and phospho-4-ABP. Whereas for Rapa-Link, I can't remember if I put the boxes here. Let's see. Yeah, so for Rapa-Link, you can see that the recovery really doesn't start till 24 hours, and then you get mostly full recovery by 48 hours. I'm sorry, I'll go back one. And for Rapamycin, there's really no recovery by 48 hours. So this argues that that MLN128 has poor residence time. It shows it doesn't have a durable connection. It doesn't have a durable binding to mTOR itself. And by adding the rapamycin moiety here, we basically can bring MLN128 to the mTORC1 complex and improve its durability as a kinase inhibitor. So another interesting aspect just shown here in glioblastoma cell lines treated 3 hours, 24 hours, 48, 72 hours is that over time, the relative growth decreases. So it's as if the drug has some sort of long-lasting effect where by 24 hours you get a response, but by 48 hours the response is better. And what we think is going on here, well, I guess this is just showing that correlates with phospho-RPS6 signaling decreasing over time. And what we think is going on here is that these glioblastoma cells are loaded with FKBP12, which I'm showing here in primary cells that we got from the Brain Tumor Research Center at UCSF. And so the Rapa-Link sort of slowly gets into the cells, but once it gets into the cells, it's binding FKB12 and stays there. So we think it actually collects over time in the cells, and you get a more durable response over time. And so this is sort of a cartoon showing that issue. So FKBP12 is top left, and FRB is where rapamycin binds, and the orange reverse triangle is where the kinase pocket is. And so for 128, the orange pocket, the triangle is just filled, so it just binds in the kinase itself. For Rapa-Link, you actually have that same binding, but in addition, you have binding through FKBP12. And for rapamycin, you're just binding through FKBP12, and you don't actually have affinity for the pocket itself. And so if that's true, then you would argue that Rapa-Link and rapamycin should compete for substrate, whereas MLN128 and rapamycin should not, and that's true. So if you use Rapa-Link in blue on the left side here, and a dose response, you see that by 1.5 nanomolar, you're at maximal antiproliferative response. If you add rapamycin, you improve the antiproliferative response at very low dosing, but at intermediate dosing, they compete with each other and the curves cross. In contrast with MLN128, the red curve shows the antiproliferative response in the absence of rapamycin, and the green curve shows it in the presence of rapamycin, and it's additive throughout its course. And mechanistically, by Western blot, if you look at the top bars here, you can see that when we add Rapa-Link, we get a dose-dependent decrease in phospho-4-ABP1, and when we add rapamycin at higher doses, now we are losing that inhibitory activity shown by recovering phospho-4-ABP1 levels. In the bottom blot, you can see that rapamycin does not similarly compete to block 4-ABP. So on the left side, you're getting a dose-dependent blockade of 4-ABP, and that's mirrored when you add rapamycin. So these data are consistent with rapamycin and Rapa-Link competing for FKBP12 binding, but MLN128, of course, doesn't care about FKBP12 binding. So another, I think one of the last data slides I have is to look at levels of FKBP12 in a pull down. So we immunoprecipitate with mTOR and we blot for FKBP12, and what you can see is that Rapa-Link brings down a lot of FKBP12, whereas rapamycin doesn't. And a lot of people have told us this experiment never works because rapamycin doesn't really seem to bring it down. It's a bit obscure why that is. If we add MLN128 to Rapa-Link, it doesn't affect the levels of FKBP12. But curiously, if we add MLN128 to rapamycin, we now see more FKBP12 pulled down than with rapamycin or MLN128 alone. And if we add Rapalink and Rapamycin together, we get less consistent with the two competing, and with Rapamycin having poorer binding. So a few conclusions. I hope I've shown you that mTORC1 and phospho-4-ABP1 are robust targets in glioblastoma. Our studies suggest mTOR is a central target, and more specifically that 4-ABP is a central target. The upstream inhibition of the pathway doesn't really block mTOR, and the first and second generation drugs also fail to block mTOR. The first generation failed to block 4-ABP1, and the second generation block it in vitro, but just are poor pharmacologic agents. So I would guess that they won't fare well in the clinical trials in glioblastoma patients that are currently ongoing. The third generation mTOR kinase inhibitor Rapalink 1 is potent, and I didn't show you this, but it blocks mutationally activated alleles of mTOR. The FKBP12 Rapa complex has high binding affinity to TOR kinase, which contributes to both potency and durability, and these agents get across the blood-brain barrier, block TORC1, and drive regression, at least initial regression, of glioblastoma xenografts. And I think I've mentioned people as I've gone along, so I'll stop there.
Thank you, Dr. Weiss. Our audience has been asking questions throughout your presentations, so let's get started with the Q&A session. Dr. Postovit, do you know what environmental agents cause ectopic nodal expression?
That's a great question. As I've shown, hypoxia seems to be one that increases the expression of nodal. In addition, other stresses, including chemotherapy itself, seems to induce nodal expression. So I think I answered that.
Yes, thank you.
Yeah.
Dr. Weiss, we had a question for you about your methods. Could you just give a brief repeat of how you treated these tissues? Were they pre-treated, co-treated? Any details?
How I treated what?
The brains.
So how I treated the mice?
Yes.
So the dosing of rapamycin and MLN-128 was based on existing literature. It was daily dosing and standard dosing. The dosing of, so that was daily. The dosing of Rapalink, the animals couldn't tolerate daily dosing. When we went to every 5 days, they were tolerant, but they weren't gaining weight, and our vets were getting upset with us, so we went to every 7 days. So the Rapalink, which is a pretty big molecule, I think it's not going to be suitable for oral dosing. Rapalink we dosed these all as IP dosing. I think Rapalink, the clinical development of Rapalink and derivative compounds will require IV dosing and not be amenable to oral dosing. But the others, I think, can be dosed orally. We didn't, just to be parallel. But we were dosing the Rapalink once a week in the end, and we dosed the other drugs every day.
Thank you, Dr. Weiss. As a follow-on question to that, would the high binding efficiency of Rapalink be a concern for human use?
The high binding to TOR. So I don't know. I mean, on the one hand, the animals tolerate it, so that's good. On the other hand, the reason they weren't gaining weight might relate to the blockade of TOR in the brain. As I showed in an early slide, that TOR is a regulator of satiety and hunger. And so, a really potent TOR inhibitor might actually have as an on-target effect blockade of, making you not want to eat. At some level, that's much harder to deal with in a mouse because you have to, mice have to eat to survive, whereas in a person, I could probably convince you for a week or two when you're not hungry to eat, or I could give you IV alimentation. I mean, I think it's an easier problem to get around, but I think that is a theoretical concern. We still need to figure out if that's an on-target or off-target effect.
Thank you, Dr. Weiss. Dr. Postovit, based on the nodal notch feedback loop that you described, what is the naturally occurring inhibitor of nodal?
There are several naturally occurring inhibitors of nodal. The one with the, I guess, greatest affinity are called the lefty proteins, lefty 1 and 2, or A and B. And these, interestingly, are also targets of nodal. So in normal development, nodal would induce the expression of lefty, which would then be secreted and then restrict nodal signaling. However, in cancer, the lefty loci seem to remain relatively silenced. So they're methylated and really don't get expressed. So very potent inhibitors of nodal called the lefty proteins.
Thank you, Dr. Postovitz. This question is for you as well. Given nodal's instability, what methods do you use to preserve the molecule for analysis?
Oh, that's a great question. So nodal, in particularly active nodal, is unstable when it's secreted. So we would use a carrier containing a formulation. When we're using the recombinant protein as well, we actually use it fresh. So we have an assay in the lab to ensure activity. This is just a simple reporter-based assay that we utilize, and in actually a teratocarcinoma cell line. And so that's how we ensure stability. It is a very unstable protein.
Thank you, Dr. Postovitz. We have time for just one more question, and this question is for you, Dr. Weiss. Is mTOR signaling responsible for the growth of most primary brain tumors?
I'm not sure how to answer that. We've only looked at a half a dozen lines, but in our hands, it does appear to be a central regulator. The TCGA has looked at hundreds of brain tumors, and as I mentioned, about 80% of them show activation of that pathway at some level. Our data suggests 40BP is the central target, but I don't know that I can generalize based on the number of lines we've looked at.
Thank you, Dr. Weiss. Unfortunately, that's all we have for today. If you have any further questions, please consider reaching out to our speakers directly. Their addresses will be shown on the screen. As a reminder, the webinar will be archived on the scientist website, and you'll receive an email notifying you when the on-demand webinar is available. I'd like to thank everyone who took the time to join us today, and particularly those of you who shared your questions and comments. On behalf of the scientists, I'd also like to thank Dr. Postovitz and Dr. Weiss, as well as our webinar sponsors, Rockland Immunochemicals, BioLegend, Mettler Toledo, and R&D Biosystems. Thank you, everyone, and goodbye.